Hello and welcome to Nikolai's genetics lessons. In this video I'm going to talk about how genetic mutations affect DNA and RNA and protein production. And as usual, I recommend you to stop video here, read both questions, try to solve both problems, choose your correct answer, and when you would be ready you can run video again and you can compare your answer with my answer and explanation. So the first question is, one codon of an message RNA reads GUA, which specify valine. If mutation change the first nucleotide of the DNA coding for this RNA to an A, use a genetic code to determine what amino acid will be put in after the mutation. So what we have here, we have message RNA that reads GUA. So let's list this trinucleotide. This is going to be GUA. So what would be corresponding DNA sequence? This is RNA sequence, and DNA sequence would be G would base pairs with C, U would base pairs with A, and A would base pair with T, or thymine. So if we change in DNA, we'll change first letter, that is cytosine, to adenine. We are going to get new sequence. So new sequence would be A, T, and new sequence of the message RNA would be U, and A. And now we need to use a codon table, and here is a codon table. And as you see, our original sequence were GUA, and here the sequence GUA, it codes for valine, and new sequence that we got is UAA, and we can find this sequence. So first letter here, second here, so UAA here, this sequence, and this trinucleotide codes for leucine. And here's our answer. So second question, and second question is, why do genetic mutations often lead to disease? And answer A, protein can no longer be synthesized from the DNA, and this is not true, because as you see, according to this table, mutations usually leads just to different amino acid if those mutations would be in the first and second position. Mutations that in the third position usually doesn't change amino acid. Still, this is going to be same amino acid, so such mutations we call silent mutations. And actually, if we change one amino acid with a different one, sometimes it doesn't affect protein itself, because proteins usually consist of thousands of such small residues, and what actually can cause interruption in protein synthesizing when we have such a mutation that change trinucleotide to stop codon. So we have 1, 2, and 3 stop codons, UAA, UAG, and UGA. And if at the beginning of the protein normal sequence that codes for the normal amino acid would be changed to sequence that codes for the stop codon, of course this protein would be non-functional. But as you see, there is only very small variants of such mutations, and most of the mutations may just affect how protein is behaving, its three-dimensional shape. But if such mutation doesn't affect active site, because protein can be very large, but active site can be rather small, and if mutation doesn't affect this active site, such mutation also can be silent. So we can cross out this answer, and next answer would be a change in amino acid sequence can change protein shape. This is true, and let's check answer C. The mutant DNA itself directly interferes with the organism normal functioning, and this is not true. DNA code for the proteins, and if there is any changes in DNA, then misshaped protein can be produced, or it can be interrupted if stop codon would happen instead of normal sequence that code for the normal amino acid. So answer C is not true, and correct answer would be answer B. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any, and see you in the next video. Goodbye.